All right, and our last home run hitter is Dr. Mark Wiesner. Now, Dr. Wiesner also has won the Clarke Prize, and he did it in 2011. All right, so he's the James L. Merriam Professor of Civil and Environmental Engineering at Duke University, and he's conducted a huge amount of research over the years on membranes. I most know him for his long-standing excellent research and leadership in membranes, but he's also worked a lot on coagulation and nanomaterials, and he's going to talk to us about nanomaterials here. He is currently, let's see here, I want to get this right here. He's the director of the Center for the Environmental Implications of Nanotechnology at Duke University, and he's got a bunch of other nanomaterial projects and centers going along with it. I'm sure he'll mention some of those. But Mark, please begin.
Thanks, Bruce. Well, if we're going from cleanup hitters to whatever, I hope home run. Well, cleanup, I was thinking I'm going to leave a mess. So let me start by thanking the organizers for the invitation to present here today, and again to pass on my congratulations to David Sedlak for the Clarke Prize. It's a great thing. How do I get rid of that little click on comment? Do I just click on it? How do I get rid of that?
Exit.
All right, good. Okay, so is it gone? I think. Oh, are we not in full screen?
Oh yeah, hit the full screen. We lost
Control
full screen.
L. Is that better? Good. So, yeah, last year I spoke about applications of nanomaterials to some technologies for water treatment, membranes in particular, and this year the sense was that I should talk about the work that we've been doing looking at the implications of nanomaterials. So I can't possibly squeeze in everything that's going on in the center and some of the other projects that are going on, but I'm going to just scratch the surface, give you a sense of what we're doing, and perhaps it is at least as important as that is to give you an idea of kind of the thinking that's going on behind some of the work that we're engaged in. Before I go further, let me just say there's a lot of people that have been involved in this research. This is my research group. Sorry, I got the green one now. Got the research group here, and these are some of the people particularly involved in the work that I'll present today, particularly those that are in bold. But then some of my colleagues in Saint, and I want to really highlight Greg Lowry at Carnegie Mellon, Mélanie Olfin at the Geosciences Environment Lab in France, as is Jean Bottereau, Jason Unrein at the University of Kentucky, and Christine Hendren, who's she's the executive director of Saint, and Greg is the deputy director. So some of their work will be highlighted here. So when I first started thinking about this problem, and it's been 15 years now, and was writing a proposal for a center that we ultimately got at Rice University, looking at applications of nanomaterials to environmental engineering, but also I put in there a part on the implications. And the way that experimental matrix was set up was basically in this fashion, where the idea was you have nanomaterial properties and you ultimately want to map those things into risk. And I think this has been more or less the approach that has been followed since that time, where you want to look at things like composition, band gap, size, etc., and see how that might affect things like exposure or hazard, whether the hazard be at the organismal level or all the way up. Now we're looking at things at the level of ecosystems. It's a bit more complicated than that. First of all, there were some efforts early on at OECD, my laboratory, and many others participated in that, where you'd go off and measure nanomaterial properties and look at how those properties, ultimately the hope was we'd be able to see how they affect risk. But what are you measuring as nanomaterial properties? Well, there was some confusion about what is really a nanomaterial property versus what are the properties of a nanomaterial in a system. So the system will affect the properties, and one needs to be clear about that. So my favorite example of that is the zeta potential or surface charge, which is very much a function of the environment that particle finds itself in. There's also the properties of the social system, the way we use nanomaterials, the trajectory of commercialization, the quantities that are produced, and so on. All of these things combine also to help determine things, certainly like exposure. You can see that very clearly from the production quantities. So we need to think about these social systems and the environmental systems or other systems. It could be a polymer matrix that some of these materials find themselves in. That whole notion of trying to map from nanomaterial properties down to risk, we haven't really been able to do that with much simpler materials. And Rhodes Trussell's presentation this morning, I think that really resonated with me. He was addressing this issue of basically how does one predict, in that case it was more on the hazard side, but ultimately risk associated or calculated from fundamental properties of materials. And we need actually, I would claim, to make any progress and get useful answers for the midterm, we need to have some sort of functional assays, is what I'll call them. Things that we can measure that will give us useful quantities that we can use to calculate something about exposure or hazard. And the best example I could give perhaps to this group is BOD. Okay, if we had looked at the problem of oxygen in surface waters back at the beginning of the 20th century and said, you know, to figure this out, we really need to understand what are all of the details of the microbial flora that's out in wastewaters and in surface waters. What are all of the properties of the carbonaceous material that it might degrade? If we had taken that approach to trying to figure out how much oxygen is going to be consumed, we would have never really gotten through the progress that we saw in the 20th century. So we developed this functional assay, this idea of measuring how much oxygen is consumed in a somewhat realistic system that can be generalized to a useful degree. So that's what we want to do here. And if you sort of think about that from the standpoint of a mass balance and think of from an exposure standpoint, nanomaterials and their concentration and how that might change over time, a lot of the functional assays I would suggest that one would need are going to be related to rates of various reactions or processes that will occur in that system. So things like aggregation or dissolution or biouptake, these are all things that we need to have rate data on in order to calculate properties of exposure and so on. So thinking about biouptake, for example, this is an image that I am very fond of. It came out of Paul Birch and Jason Unrun's lab at the University of Kentucky. And what you see here are gold nanoparticles that have been taken up by tobacco plants, and they're distributed through the leaves. So when I was in graduate school, I think even today, the very notion of measuring suspended solids is you take water, you put it on a 0.45 micron membrane, and everything that's retained we call particulate, and kind of by implication, everything that went through is maybe functionally dissolved. But of course we know it's not. And with nanotechnology, we can now characterize using electron microscopy and many other techniques, hyperspectral imaging, we can begin to characterize the materials that would go through that membrane. And we understand that it's a very rich speciation of materials. Iron, for example, that goes through is not just dissolved iron. You might have some truly dissolved ionic species of iron, but you might have iron of all sorts of different cluster sizes, and those different sizes are going to play different roles. Plants, of course, have known about this, as have other living things for a long time. They use these things, and we'll come back to that later. So plant uptake, it occurs, and we can think about doing some functional assays to determine the rates at which that occurs. Dissolution is another example. So here's an example I'll give you from Peter Weichsland's work at Virginia Tech. So dissolution at the nanoscale, he's developed a nice method where he can deposit systematically basically particles of varying size and geometry on a surface and explore the effect of those parameters on dissolution rates. So you can get dissolution rates from that, a sort of functional assay that could be applied in a more complex system. Here's one that a bunch of us have worked on for a time, but Greg Lowry has really spent some time with this, as is Helen Shi Kim, myself, Melanie Olfan. The idea of sulfidation, so transformations that occur. The nanoparticles that come out of the box aren't the ones that you're going to see in your bloodstream or that you're going to see in the environment. They're going to change. And so sulfidation of some of these soft metal nanomaterials is particularly of interest. We spent a lot of time thinking about silver and a bunch of time thinking about zinc and copper. Copper is probably the more recent one we've been looking at. So here's an example of why this makes a difference. This going back to plants. So these are X-ray fluorescence images of uptake of silver in 3 different forms, either supposedly ionic silver. I say supposedly ionic in the form of silver nitrate because in the presence of light and some macromolecules, there's actually a pathway where metallic nanoparticles can be formed. You can do this. There's all sorts of interesting things that have been published using green tea, for example. But some uptake involving silver nitrate, and in this case, it basically just killed the plant. Silver nanoparticles themselves, lots of uptake going on, less lethal to the plant. And then in the case where we sulfidize it, there's still uptake going on, but as you'll see, much less of an effect on the plant. We've done this not only for the plants, but we've looked at a few animal models, so zebrafish, fungulus or killifish, C. elegans, a worm. And in all of these cases, these are LC50s you see going along here. And as these numbers go to the left here, we're going into less and less lethal territory. So sulfidation, take-home message, sulfidation decreases the toxicity in all of those models. So that's information that's come out of some of the laboratory studies that we've been engaged in, and we want to look at more realistic scenarios as well. Sulfidation we've observed happening in a mesocosm facility. This simulates a freshwater wetland that we have. It's out on the Duke campus, the Duke Forest actually, and wastewater treatment as well. We've looked at that in both benchtop and then in pilot scale facilities. This is with some collaborators of ours in England in a sort of a paired study along with Saint, where we were looking at taking a slipstream off of the wastewater treatment plant, treating it, putting in either, again, ionic zinc or zinc nanoparticles, running it through all of the motions of aerobic treatment, anaerobic treatment, ending up with the biosolids, doing that centrally at one place, and then taking those biosolids and distributing them throughout all the partners and looking at the impacts of those materials and the transformations that occurred. I'll say a little bit more here about the mesocosm system, so just another picture of that. This facility is designed to do longer-term studies. They run typically about a year. Some of them are multiple boxes. Each one of these mesocosms, there's 30 of them. They're about a meter high, meter wide, 4 meters long. And we've run all sorts of things in them. So we've done it, nanomaterials using pulse inputs, chronic inputs, nanosilver, cerium dioxide, copper. We're doing some things, particularly now copper and cerium dioxide release from commercial wood products or stains that go on wood. Nanomaterials with some conventional contaminants and single-wall carbon nanotubes and others. So we've run a few different things. One of the things that we've observed is when you run an experiment in the lab, at least the first time, so take an organism like the zebrafish, the killifish we've had out in these mesocosms. Take the killifish in the lab, create conditions that you think are similar to what you're going to have out in the mesocosms, and we don't necessarily see the same thing. Sometimes the toxicity has been more, sometimes it's been less. Why? Well, it has to do with things that you didn't really bring back to the lab, that you didn't think about the first time. In this case, it had a lot to do with, again, killing plants, releasing organic matter, and then transforming the actual properties of the nanomaterials that are present in these mesocosms. So trophic transfer. We've looked at this in a few different scenarios, from bacteria to the C. elegans, the worm model we use, or from tobacco plants to the tobacco hornworm, which actually we've taken that a few steps actually to frogs as well, and some other cases. Now, what you see, we've observed trophic transfer. In some cases, that trophic transfer is accompanied by biomagnification. The concentration of this material increases as you go up the food web. So that was the case with the tobacco hornworm and the tobacco plant. In other cases, you actually get a trophic dilution of the material. Okay, same nanoparticle here, a gold nanoparticle that was present in smaller concentrations in the tissue of this frog than, for example, the tissue in the previous trophic level. So that gets us thinking about how do we predict these sorts of things, how do we model it, and what are, again, these the appropriate functional assays that we would use to describe these processes. If we were doing this for the case of conventional contaminants, and I think it's great that I'm ending this today where Urs had started out, he even mentioned nanoparticles at the beginning, but started out talking about some of these conventional contaminants. If you take some of those things and think about their KOW, what's their potential for partitioning between water and an organic phase? Well, the KOW is a nice predictor not only for bioconcentration, where you might imagine a fish, for example, to just be basically a swimming sack of lipids, and so the more that material can accumulate in the lipid phase, that should be predictable then from KOW. But we can also think about it from a transport stage. We use this all the time in groundwater transport to predict the retarded velocity of transport of a contaminant moving in the groundwater, where if you have a higher fraction of organic carbon in the groundwater and the aquifer material, that will tend to absorb materials as well. And we can correlate that with the octanol water partition coefficient. Well, so what would be the parameter like that we would want to have for nanoparticles? And I want to suggest that this thing, it goes by many names. Surface affinity is what I tend to use as a term for that, but many of us know it lovingly as alpha, or the attachment efficiency. And it has to do with the importance of heteroaggregation and deposition in the fate and transport of these materials. So, of course, we can talk about auto or homoaggregation between particles, just how sticky are they with respect to one another. But then how do those materials begin to interact with some of the other surfaces that they might see in an environment? In our mesocosms, we might have a frog skin surface, or we might have something much better defined like a given mineral surface. So evaluating quantitatively using a functional assay the affinity of a nanoparticle for those various surfaces, perhaps in an ideal world you could even do that with some reference materials. Just like KOW, we could have used olive oil and water, but we have a nice solid reference material in octanol that correlates quite well with olive oil water partitioning. Well, maybe we can have some reference surfaces that would also be of use to us, or we take the much more, the dirtier, more complex approach of looking at their affinity for some of these more complex surfaces. Autoaggregation, a bunch of reasons it's important to us. First of all, it affects everything from the apparent nanoparticle reactivity. It affects things, Vicki Grassian at Iowa, John Lanehart at Ohio, and others have looked at the role of aggregation on dissolution. But David Jasby and Matt Hotzig and my group have looked at how when these surfaces get together, surfaces are reactants that in an aggregate or in higher concentrations than they are if they're just simply spread out throughout the suspension. So you can you want to look at how those effective concentrations change as a function of aggregate size and morphology. And so that turns out to be important for things like C60, singlet oxygen generation. Pedro had presented a little bit of that in the disinfection application of that earlier. And you can do that for semiconductors like titanium dioxide and others. So we go to our mesocosms. We want to predict what's going on in a mesocosm. We can measure this. This is some data that actually came out of, actually this is a simulation, and you can hardly see it here, but the data, which the two look quite similar. We model the fate of nanomaterials, in this case, you see them dropping off very, very quickly in about a week's time, and that's turned out to be the case with almost every nanomaterial that we put in a pulse scenario. It drops very quickly. We want to model that. The decreases in concentration are very closely associated with the heteroaggregation that goes on. We've taken models that are based on aggregation theory that have to be expanded for the heteroaggregation case, so you're tracking multiple materials that are interacting. And you end up with simulations kind of this scenario here where you put in this case, a bit of nanomaterials into a system where there's some background particles, and those nanomaterials begin to interact with the background particles, and very, very quickly those nanomaterials are associated with the background material. So you can think in terms of a half-life of a nanoparticle being just an independent pre-nanoparticle as opposed to being associated with some of this background material. Do calculations of those half-lives for a whole bunch of different conditions that would be potentially plausible for the aquatic systems that we see. And what you take away from those simulations is that heteroaggregation is basically going to move things into contact over the ranges of concentrations and sizes of materials that we're going to encounter. It's going to very quickly move things into contact with those background particles. And those background particles are then, that's what you want to track. Those are what are going to move through the system and determine the fate of these nanomaterials. All of that, being able to model that, depends on being able to quantify, again, going back to this functional assay for surface affinity. How do we quantify surface affinity? Well, you could take the material that you wanted to see that particle sticking to and put it in a column and run it through the column, and using some filtration theory, get an estimate of the stickiness between that particle and the stuff in the column. We've developed another method for much more complex systems where you take materials, you can mix it for programmed mixing times, and then separate the nanomaterials, or separate the aggregated materials from any nanomaterials that remain unaggregated. And you can plot then the distribution of basically heteroaggregated and free nanomaterials versus the time at which you gave them to get to know one another, basically. And there's a linear relationship that occurs during the early stages there that can be used to extract the affinity coefficient alpha. And it's also proportional to the concentration of background particles and to the collision rate kernel that determines how quickly these things are coming together. You can either get those things out by measurement and or theory, or you could take conditions that would favor aggregation, sort of like we do in column studies for those of you that do these things, where you make alpha 1 and then you basically get a fitting parameter that determines what the beta B term would be. So bottom line, we can get alphas from this, and some of the more recent results that we have that really very encouraged by is when we measure alpha in a column, and we measure alpha using this sort of a system, and we do all the things where alpha should change, changing pH, for example, of the system, changing ionic strength. We get very nice correlations between those two methods. So back to the effects, okay, and this is really a schematic of how I might think about a nanomaterial reacting with a given surface. So you have a nanoparticle out here in the surface it's going to react with. It could be a bacterium or a virus that it's going to inactivate. So the nanoparticle to go through that inactivation, let's say that we're going to do disinfection as Pedro had suggested. We want to move that nanoparticle up to the surface. We need to know something about the transport. The attachment then tells us whether or not it sticks to the surface. With something with a very short lifetime as say a singlet oxygen, it has to be on the surface to deliver that and get inactivation. So then the reaction occurs. If you observe the reaction from a distance, what you're observing are the sum total of all of those steps. okay, as opposed to the intrinsic reaction rate on the surface there. So stickiness makes a difference, and I'm just going to give you a few examples before I finish. So here, again, to get back to our plants, here are plant roots that have been exposed to just deionized water, the control, but particles that have two different kinds of coatings. In one case, gum arabic, in another case, PVP, and then this is a silver nitrate. You can see, without getting into the details, that the surface chemistry makes a difference. This is the case of cerium dioxide reduction. Lauren Barton's work, who just graduated from my group about 6 months ago. She was looking at the fate of this material in wastewater treatment. So, ironically, in an aerobic system like an activated sludge tank, there's a lot of reduction going on of cerium dioxide. There's also the reverse reaction where the reduced cerium dioxide is reoxidized. But you can follow this. These are in Zane's spectra that we obtained where this peak, this is the spectrum for cerium-4, and this is cerium-3. And then you can follow the increase in the cerium-3 peak to get rates of cerium reduction. And what's different between these two is that in the one case we have a bare cerium nanoparticle, and the other one it's a citrate stabilized. So again, differences in surface chemistry, differences in rates. Viral inactivation. Okay, so we did this work quite some time ago looking at hydroxylated fullerol, or C60 in this case, and its interaction where here the surface that's changing is not the surface of the nanoparticle, but it's the surface of the virus that it's interacting with. And when there's more affinity for the surface, you have more association of those nanoparticles on the surface. You hit them with light, they produce singlet oxygen, you get greater degrees of viral inactivation. And again, another case, this is the killifish, a nice model because it's an estuarine species. It's tolerant to both salty and fresh water, so we can change things that we like to change in colloid chemistry, like salt concentration. And much of the toxicity that one observes can be predicted by this gray area here, which is just equilibrium chemistry for silver, but not all of it. Okay, there are differences that occur when the nanoparticle has a gum arabic, a PVP, or a citrate coating on it. So again, you can model this thing. The way we modeled it is where you can think of the transport attachment step as one resistance in a series of resistance. So that's one resistance along the reaction step, and then the actual reaction itself is the other resistance. So it's resistance in series, and that leads to an expression like this, where you have an overall reaction rate, Kt, that is a function of the intrinsic reaction rate, here shown as K red, and then some of the heteroaggregation parameters, in particular this surface affinity alpha that we can get out of that functional assay I showed you where you're mixing materials. You go back and you look at Lauren's data where she was able to measure these surface affinities in very complex systems, like activated sludge, and it turns out that the differences in the rate constants overall observed are exactly predicted by the differences in alpha for that system. So I'll just leave you with this, the idea that what we're exploring now is that the surface affinity is going to be, we want to know if it's going to be a predictor of things, in particular biouptake in something more complex like a fish, or how that affects the uptake and translocation of nanoparticles in plant species. So I think we need functional assays to get really off of dead center and moving forward to do calculations of exposure and hazard. I really didn't hit on it too much, but this idea, when you see particles moving through these plants, and the notion that plants and other organisms are using nanoparticles in a fashion that it doesn't matter if these particles are toxic or not. That's been much of the focus of what this work has been, is the idea that it could be an emerging contaminant. But I would claim that it really, the importance of this is how living things interact with these nanoscale objects. And it goes from everything from the way they might obtain nutrients, to the way, to again cite an example that Pedro highlighted, where we've hypothesized that small genetic material, other macromolecules can sorb onto nanomaterials, move through systems, and so on. With that, thank you for your attention, and time for dinner.
But before dinner, we may have a question or two for Mark. Let's see.
Everyone wants.
There's one. Oh, Bruce has got one.
Well, we started off the day talking about how chemicals might or might not make it through wastewater treatment plants, and you know, it kind of sounds like, given heterocoagulation and adsorption and all these processes that occur in an activated sludge plant, you would sort of think they'd never make it out, but they do.
They do, yeah. But the vast majority, as predicted by that, the vast majority do end up in the sediments. And then where do those biosolids go? So we have a whole effort. I've got a student looking at land application of biosolids that come out of
out of the digester, out of compost,
yeah,
and then into the lab.
Right. So, and then you want to model that system. You want to look at leaching. You want to look at plant uptake. So then it becomes a food issue.